Hi everyone! Today we are going to present about culture of specific cells. Hematopoietic cells. Hematopoietic cells is a cell isolated from the blood or bone marrow that can renew itself and differentiate to specialized cells. The sources of hematopoietic cells are from the bone marrow, peripheral blood, umbilical cord blood, embryonic germ cells, and embryonic stem cells. Basic technique. First, remove the uterus from pregnant mice into a tissue culture dish containing PBS FCS. Then transfer the embryo to other culture dishes containing PBS and FCS to wash away maternal blood contamination. Next, dissect aorta gonad mesonephros region from the embryo proper. Place wire mesh support to each well of a 6-well culture plate filling with 5 ml of myeloid long-term culture media and place a filter onto the mesh support and adjust medium level. Place the individual dissected embryo tissues on the filters. Place culture plate in 37 degrees Celsius and 5% carbon dioxide incubator. Tissue explants are cultured for 2 to 3 days. Application. First, generation of large numbers of mature blood cells or lineage-specific cells. Second, the expansion of stem cells capable of reconstituting hematopoiesis after transplantation. The third application is genetic modification and last but not least, identification of novel regulators of hematopoiesis.
Next is gonad cells. Gonad is an endocrine gland that produces the gametes, germ cells of an organism. The male gonad testicle produces sperm while female gonad ovary produces egg cells. Basic technique. Firstly is preparation of beta agar with DMEM, then preparation of agar block for the organ culture of gonad. After that, collect embryos from wild type or transgenic mice at the developmental stage of interest. Then, reserve a bit of each embryo still for sex confirmation by PCR. Next is dissect the entire bilateral urogenital complex along with the dorsal aorta from all of the embryos. Then, tissue containment in pre-warm DMEM. Keep tissues at 37 degrees Celsius and 5% of carbon dioxide. Then, separation and removal of unwanted substances or organ from the gonad organ culture by using pipette. Assemble the organ culture and remove remainder DMEM. Add organ culture medium. Next, incubation of organ culture at 37 degrees Celsius and 5% of CO2 and preservation by changing the medium daily. The application is germline research.
Now everyone, let's look at neuroectodermal cells. It is a portion of ectoderm of the early embryo which gives rise to central nervous system and peripheral nervous system. It can be found in the neurocrest, which is the pigment cells in skin, and the other one, the neurotube, in the brain, spinal cord, retina, and posterior pituitary. The basic technique. First, dissect out the cerebella and place them in HBSS. After that, mince it into small cubes, then transfer it into the test tube and wash it 3 times in HBSS. Follow with the addition of trypsin and incubation in water bath. Next, transfer the trypsinized tissue to centrifuge tube and stop the trypsinization with the growth medium. Then, disaggregate it by trituration through siliconized special pipette until we can see single cell suspension. Allow the small clumps of the tissue to settle down, then remove it. After that, centrifuge and aspirate off the supernatant. Next step, suspend pellet in the growth medium and seed the cells. After 2 days, incubate the culture with cytosine arabinocytes, and lastly, change to regular culture medium. Application: stem cell transplantation in regenerative medicine.
Created using Powtoon.